I'm excited to introduce today's lecture. Dr. Robert Innis is the chief of the Molecular Imaging Branch at the National Institute of Mental Health. Dr. Innis holds a BS degree from Yale in molecular biophysics and biochemistry. He received his MD and PhD in pharmacology from Johns Hopkins University. His research focuses on the use of PET imaging to study neuropsychiatric disorders. Please enjoy the presentation.
Welcome. My name is Dr. Robert Innis, and I'm very glad to speak to you today about my area of research, which is in positron emission tomography. And I want to explain it in terms of its principles and what it can do of relevance to pharmacology. And in particular in that regard, how PET, positron emission tomography, can be used to study pharmacokinetics and also to facilitate therapeutic drug development. What I will tell you today, I'll summarize at the beginning. And the first is to say that PET, positron emission tomography, is an in vivo technique with very high sensitivity and specificity. It can measure compounds present at very low concentrations in the body, and it can measure specific proteins labeled with radioligands. I'll explain how PET is currently used in therapeutic drug development, primarily to determine the appropriate dose to be given on phase 1 studies. I'll also try to explain in words and by simple analogies what pharmacokinetic modeling provides. And it's basically measuring the concentration of drug in the plasma concentration, and the tissue uptake. And if you do both of those, you can measure the density of the target, of the receptor that the drug is working at. I'll show examples of how PET can be used to study distribution of drugs. Distribution, by the way, is the reversible transfer of drugs from the plasma to the tissue and back. So if it's distributed, the drug goes to the tissue and then comes back to the plasma. And the specific example is one where you can block the distribution to the periphery and increase the distribution to the brain. I'll also show an example how PET can study metabolism, where metabolism is making a new bond or breaking a bond, destroying, in some ways, the original chemical structure of the drug. In this particular case, it will be in regard to inhibiting defluorination. Okay, PET has been around for several decades, going back to the late 1970s. And the general strategy is shown here. A cyclotron is used to produce unstable or radioactive atom. For example, carbon-11 can be made in a cyclotron. It has a half-life of only 20 minutes, so you have to use it very quickly. So it might come out of the cyclotron as C11 labeled CO2. And then it's incorporated into some drug rapidly, made appropriate for human injection, and then the subject is injected and is in a PET camera, which is like a sort of a donut hole, and the head or the whole body can go in. The result of that, for example, would be this image, a tomograph, where a tomograph is a slice. This is a slice through the brain, where we have the front of the brain, the back of the brain, and the two sides. And you can see that this C11 labeled drug, in this case raclopride, which binds to dopamine receptors, is present in the internal portion of the brain. So this cartoon comes from the mid-1980s, when the resolution of PET was on the order of about 16 millimeters. The resolution has gotten a lot better since then, and the pictures are less blurry. I like this next movie clip made by Simon Sherry from UC Davis, and it shows the principles of PET imaging for the most common clinical use currently. And that is a radio-labeled form of glucose or sugar is injected with patients with cancer, most cancers, many of them. And it so happens that tumors, as well as their metastases, burn up a lot of sugar and take up a whole lot. So you can localize the tumor and its metastases by increased uptake of a radio-labeled sugar analog. Okay. So in this movie clip, you'll see the donut-shaped PET camera, and we have the IV ready to inject the compound here. The radioactive F18, fluorodeoxyglucose, is scintillating, so it's within a glucose analog. It travels through the body, and then it will be taken up by a tumor in the lung. What happens with this unstable radioactive atom is that there's a nuclear decay. A particle is emitted from the nucleus of the atom, and after a certain period, that particle is a positron, and at a certain time the positron will annihilate, will meet with an electron, so that's a matter and antimatter, a positron and an electron meeting, which gives off pure energy in the form of two gamma rays which go out at 180-degree angles, and then can be detected by the PET camera and reconstructed into slices throughout the body or throughout the brain in this case. I like that because it's I'm still fascinated by this. Ever since I was a medical student at Hopkins in the late 1970s, and I first heard about PET. It was matter and antimatter combining, giving off pure energy in the form of two gamma rays, and being a Star Trek fan, I said, Scotty, bring me aboard, beam me aboard, because that's what I want to do. And it is amazing. Sometimes for students, I will ask them, if I give you the mass of the electron and the mass of the positron, I want you to calculate the frequency of the gamma rays that are emitted. And you can do that. And you have to use Einstein's equation, E equals mc squared, because the mass is converted into pure energy. It's amazing that we're able to do this now. Okay, another way to understand PET is by a comparison with magnetic resonance imaging, which is far better known. And the spatial resolution of MRI is much, much better, less than 1 millimeter for the MRI. Whereas PET, well, PET is down to about its limit now of about 1 millimeter. But the way in which PET really stands out is its sensitivity. Whatever MRI or MRS is measuring has to be present in vivo at 10 to the minus 4th molar, whereas PET has a sensitivity down to 10 to the minus 12th, perhaps 10 to the minus 14th molar. So it can see that radioactive atom, carbon-11 or F18, if it's present at very low concentrations. It turns out that many of the proteins that we're interested in the body and in the brain that might be involved in pathophysiology are present at 10 to the minus 9th molar, 1 nanomolar. So there's no way that MRI would be capable of measuring proteins at such low concentrations, whereas PET is certainly able to do so. So the radionuclide, the unstable atom, carbon-11, rather than the normal form, carbon-12, can be detected in vivo with very high sensitivity. If that is combined with a drug like raclopride, it can have high selectivity for the D2 type of dopamine receptor. You end up with a radioligand, C11 raclopride, that has high sensitivity because of the carbon-11 and selectivity because of the raclopride. So this can be used for any protein in the body, enzyme, receptor, transporter, and be able to measure it in vivo with a PET device. So a radioligand then is a drug plus some radioactivity with it. And I should mention that the drug is administered at tracer doses, at minuscule doses that will have no pharmacological effects. And we can administer such low doses because PET has the sensitivity to measure it in the body after it's distributed to the body. Because it's a tracer dose from a pharmacological perspective, there are no pharmacological effects. It might bind to or label, less than 1% of the receptors. But that labeled subset reflects the entire population. So you use the radioligand to label a representative subset, perhaps less than 1% of all the receptors, in order to study those receptors. As a drug, the radioligand is disposed of in the body, where disposition in this case is referring to, for example, metabolism and distribution. Metabolism is where you break a bond or add a new bond. And distribution refers to the transfer of a drug from the blood, plasma, to the tissue and back. And I'm going to show you examples of how PET can be used to study metabolism and distribution. The major disadvantage of PET is that there is radiation exposure associated with it. And the radiation exposure is a very complex and controversial. But I think it would be fair to say, based upon large numbers of studies, particularly the survivors of nuclear bombs in Hiroshima and Nagasaki, that the level of radiation that we have here has never been documented, for example, to increase the cancer rate. So it would be considered safe within our current knowledge. And it is currently used very commonly, for example, in cancer patients where you have the radio-labeled glucose analog. But it is associated with radiation exposure. Okay. Well, I was mentioning that the resolution has gotten better, and that's shown here in this slide, a movie clip. So in this case, this was a rat that was injected with sodium fluoride, the F18, the unstable isotope, not F19. And fluoride, as you may know, goes to the bone, particularly to the growth portions of bone. So you can see really the level of detail that you can get here when you inject a rat intravenously with sodium fluoride, and the F18 is taken up particularly in the growth portions of the bone. So the resolution has gotten a lot better. We can image rodents, mice, transgenic mice, not just large humans. Okay. So I want to tell you examples of how PET can be used and is being used as a tool in therapeutic drug development. And by far the most common use is to determine the dose that should be used and the dosing interval. If you have a new therapeutic drug, how much do you have to give, and how often do you have to give it? A good example of that comes from this slide. I think this must be from the early 1990s from Brookhaven National Laboratory, Joanna Fowler, Nora Volkow. And it looks at the uptake in the brain of a C11 labeled Deprenyl, so a radioactively labeled drug. This is actually an inhibitor of monoamine oxidase B, an enzyme in the brain involved in the metabolism of monoamines. So the subject here was scanned on 4 different times. One at baseline, and you can see high uptake, high amounts of monoamine MAOB in the brain. And then after giving a blocking dose of lazabamide, which is an MAO inhibitor. So this is non-radioactive lazabamide. You can see what happens, how you block the uptake at 25 BID, 50 milligrams is more, but that it all returns after about 36 hours. Okay, so let's compare this to if you used selegiline, which is a marketed antidepressant and also an MAOB inhibitor. You can see baseline here, and then 5 milligrams BID. Wow! So it's clear that selegiline is more potent than lazabamide because 5 milligrams did more than 25 did. But perhaps even more striking is the fact that this blockade of the enzyme appears to last for several weeks. Well, the reason for that, by the way, is that lazabamide is a reversible inhibitor. It binds to MAOB and binds and comes off. And when it's binding, it blocks the binding of the radioligand C11 labeled Deprenyl. In contrast, selegiline is an irreversible inhibitor. It's a suicide substrate. When it binds to, it also covalently binds to MAOB. So what we're seeing in the bottom slide is actually the production, the synthesis of newly made MAOB, replenishing that had been irreversibly inhibited by selegiline. So I think that this sort of schematically shows how you can use a radiolabel and looking at therapeutic compounds to find, well, how much are you going to have to give and how often do you have to give it? For the latter question, for example, I mean, there'd be no reason to give more than 5 milligrams twice a day of selegiline because it'd have a complete blockade. And there's no reason to give it every day because the effect lasts for at least a week. And so this is commonly done now when a new therapeutic drug is being considered, particularly for the brain. And it's a very important question. You have a new drug, it acts at receptor X. How much are you going to give in the initial study? Do you even know that the drug passes the blood-brain barrier and makes it into the brain? So this is a very important question. If that dose is too much, you could have a lot of toxicity. Well, unnecessary toxicity if you gave more than was necessary. But if you gave too little, well, then you're going to have no efficacy. You think it failed, but it really wasn't that it failed, it's just you didn't give the right dose. So this can be like a $200 million question. And you can use the radiolabel compound to say, for example, with this compound, I think 25 BID is more than enough. And I think we should give it, you know, maybe even once a day. But it'll give you some rough guidance to it. And if the 25 milligrams BID does not treat the depression, you can say, well, this drug is ineffective at acting at this target to treat depression. And you really know that it's a negative study. Okay, so that is commonly done in most pharma companies that I'm aware of now, at least for CNS drugs, central nervous system drugs. If they begin to look at a new target, right at the time that they identify some new target that they want a therapeutic compound for, they allocate resources, money, people, supplies, to also make a PET radioligand for that target so that it is ready on the initial human studies to determine, did the drug actually get to the brain? Was there target engagement, as that's the term for it, and has enough gotten there? Is there a high enough receptor occupancy? Maybe 50% you'll be looking for, but there's no reason to go above 100%, and there's no reason to go then less than 0%. Another way that PET can be used is to identify a more homogeneous group of patients. And the example that I'll give here and in the next one has to do with Parkinson's disease. This cartoon shows a synapse, or the connection between two neurons in the brain. And the presynaptic terminal contains the transmitter, in this case dopamine, in vesicles, which are released by exocytosis, diffuse across the synaptic gap to the postsynaptic site and might interact with D2 dopamine receptors, for example. The signal is terminated by dopamine being taken back up into the presynaptic terminal where it can be recycled or metabolized. The way that cocaine works, or at least one of its mechanisms of actions, is by blocking the dopamine transporter as shown here, thereby leading to elevated concentrations of dopamine in the synapse, which are thought to mediate the high from cocaine and also its addictive properties. Well, the connection to Parkinson's disease is that for unknown reasons, there's a degeneration of the dopamine-containing neurons in the brain. You lose the dopamine terminals, you lose the dopamine, and you also lose the dopamine transporter. And for that reason, several years ago when I was at Yale, we made a radio-labeled analog of cocaine in order to measure the cocaine receptor, the dopamine transporter, because we expected it would be reduced in patients with Parkinson's disease. Okay, this shows an image. At that time we were using a related technique. I won't go into the difference, but it's radioligand studies with a single photon emitter of the dopamine transporter. And you can see in this study that the resolution of the SPECT in this case is like about 16 millimeters, like the old-time PET. And you can see that there's high uptake, as expected, in these terminal areas, the caudate that I'm showing here, and in the putamen. So this is what a healthy subject would look like. And we quickly went into patients with Parkinson's disease. In this case, this was an individual in stage 1. Parkinson's disease is an idiopathic movement disorder. It's very bad. Perhaps a third of the patients will actually develop full-blown dementia. So it's a severe, terrible neurodegenerative disorder. And this person is in stage 1, which is when you might have symptoms where you have symptoms on only one side of the body, maybe a tremor on just one side. It has not yet progressed to hit both sides. And you can see clearly there's a significant loss of these dopamine transporters to the cocaine receptor. Actually, the symptoms begin when there's about a 50% loss. You can also tell, and this is done quantitatively, but there's a greater loss in the putamen than in the caudate, which is consistent with the known neuropathology at the time of death in patients with Parkinson's disease. There's a greater loss of dopamine terminals in the putamen than the caudate. And this also shows a right-left asymmetry. And we can be certain that this patient's symptoms, 100% of the time, began on the other side of the body from where the greater decrease was found. So there are three distinguishing features of the dopamine transporter in Parkinson's disease. There's decreased uptake, symptoms beginning at about 50% loss. It's asymmetrical, which always reflects the side of the body with the initial onset. And the loss in the putamen is greater than the loss in the caudate. So this can be very helpful in therapeutic trials because this is the really bad disease. The other alternative is where you just have a little bit of tremor in old age. It's grandma with a little bit of tremor, and that's called benign senile tremor. It's just got a little bit of tremor. It's not going to get worse. It's not going to develop into Parkinson's disease. And those people with benign senile tremor will have normal or healthy dopamine transporters. Only the people with the Parkinson's disease will have this 50% loss or greater. So if you have a drug that you think could be helpful in Parkinson's disease, you'd want to start it as soon as possible, and you'd want to eliminate those individuals who have benign senile tremor. Nowadays, I think it's fair to say that if you go to the neurologist with a little bit of tremor, they can't tell. And you just have to follow a person for 1 to 2 years. If it progresses, it's the bad disease. If it doesn't progress, it's the benign senile tremor. Well, this can be used and is actually approved to be used to aid in the diagnosis of Parkinson's disease to distinguish those two conditions. It was approved first in the European Union and first here. And it can be used to get a more homogeneous group of individuals for this putative neuroprotective treatment of Parkinson's disease. It's going to have people with Parkinsonian symptoms plus an abnormal scan showing loss of dopamine transporters. And it has been used for this purpose. Okay, in addition to identifying a homogeneous group of individuals, more homogeneous, using it as a biomarker, if you will, it can also be a biomarker for drug efficacy. And that's shown here. These are repeat images in a patient with Parkinson's disease over a period of about 4 years. And you can see that there's a relatively rapid loss of dopamine transporters in this individual. And that's true of the bad disease. There's a rapid deterioration. Well, if you have some intervention that's supposed to slow the progression, this can be used. If it's supposed to slow the progression, then it should slow the loss of this biomarker, which is on those dopamine terminals that continue to degenerate. It can be very hard to measure the progression of Parkinson's disease based upon clinical symptoms because they vary during the day and it's hard to get one measure. But this is an endophenotype, a biomarker in the brain that can be used, and that certainly should not deteriorate if the neuroprotective agent is effective. So dopamine transporter imaging has been used to assess efficacy of neuroprotective agents looking serially over time in patients. Currently, probably the most well-known or widespread is studies being done now in Alzheimer's disease. So a PET radioligand was developed for amyloid. Actually, the first one was at University of Pittsburgh compound B. And this amyloid is an abnormal protein which builds up in the brain and is associated with the death of neurons and dementia. You can see there's much higher uptake in this patient with Alzheimer's disease than in the control subject. Well, several different types of anti-amyloid therapy have been tested, like, for example, making antibodies to amyloid and see if the antibodies would remove amyloid and make the people better. In order to assess whether the antibodies are effective in removing amyloid, the patients are given serial images with this PET marker or a related one to the amyloid protein itself. So it can be used as a biomarker of drug efficacy, in this case of drugs used to treat or slow the progression of Alzheimer's disease. So far, there's no clear evidence that that's effective. But it's being looked at very actively. Okay. So in addition to PET being used currently in therapeutic drug development, the most common being to determine the initial dose of a phase 1 study, PET theoretically and practically can provide very useful information about the target of drug action. And so this is what comes under the term pharmacokinetic modeling, where you measure the drug in the plasma and concurrently in the tissue. This is a very complex area. I'm going to try to make it simple by making up this story of what it was like for me when I was a resident in psychiatry at Yale. So as many of you know, Prozac is a widely used, now off patent, SSRI, selective serotonin reuptake inhibitor. Prozac, the generic name is fluoxetine, blocks the serotonin transporter. Just like cocaine blocks the dopamine transporter, fluoxetine blocks the serotonin transporter. And it's an effective antidepressant. So this is the story that I make up. When I was a psychiatry resident, I said, well, to my professor, Professor Moriarty, who, by the way, is the literary foil of Sherlock Holmes, I said, Professor, I think that fluoxetine must be effective because I'll bet that patients with depression have too many serotonin transporters, and fluoxetine blocks them and bring them back to normal. So in order to test this, I made F18 labeled fluoxetine, which has several fluorines, and I did this PET scan, and we see much higher uptake in the patient than in the healthy subject. And if we take the entire extrapolated area under the curve to infinity, it was 32 versus 16. And I said, you see, Professor, I've proved my point. Patients with depression have twice as many serotonin transporters. And he said, well, Bob, you know, that's pretty good, but tell me, how much did you inject the subject with? Oh, well, in the patient I injected with 20 millicuries, but the healthy subject, the radiochemists were having a bad day, and they only injected 10 millicuries. And he said, Bob, you stupid fool, of course there's going to be more in the patient because you injected twice as much in the patient. Go back and do it properly. So I went back and I did it again, and this time I injected in a new patient and healthy subject. Each got 20 millicuries, but I still got the same result. And I said, now, Professor, you see, I've definitely proved it. He said, well, you're doing better, but tell me, how much did they weigh? And I said, oh, well, the healthy subject was healthy by US standards at 100 kilograms, but the patient lost appetite and was down to 50 kilograms. And he said, Bob, you stupid fool, of course the concentration in the brain is twice as much in the patient because it's distributed in half the total volume as the healthy subject. Go back and do it properly. So I did. I went back, and this time I got two normal people, quote, at 100 kilograms. They both got 20 millicuries, but I still got the same result. And I said, now I've proven it. And he said, well, this is better, but tell me, tell me a little bit more about the medical history. And I said, oh, well, the patient actually had liver disease. The patient had tried to commit suicide a couple of times and completely blew out the liver. But the healthy subject had a normal liver. And he said, ah, Bob, now, how do you know there wasn't more in the patient just because the patient didn't metabolize the drug? The healthy subject was a normal metabolizer. This is a very difficult question to answer. And this is where pharmacokinetic modeling comes in to answer this question. Okay, so I'm going to refer to something called binding potential. We want to measure the total number of receptors or transporters in the brain. And this binding potential that we're going to measure is a product of the number of receptors, the receptor density, and the affinity of the drug for that receptor. So you're going to have more uptake in some individual if they have more receptors, or if their receptors happen to have higher affinity for the ligand. In general, for almost all cases, everybody has the same affinity. So this binding potential is just measuring the density. In order to answer that last question, you have to measure, in addition to the brain, you have to measure the concentration of drug in the plasma. And this is actually arterial plasma, in the plasma that's going to the brain. So this is the parent radioligand over time. You extrapolate both of those to infinity. This might be 16 and this might be 2. And the binding potential will be equal to the area in the brain divided by the area of the plasma curve. So in this case, it would be 8. It's a unitless number that is proportional to the density of receptors. How does this correct for the injected dose? Well, these are such low doses, tracer doses, that we're in the linear portion of the curve. If you double the injected dose from 10 millicuries to 20 millicuries, the concentration in the plasma will go up twofold, from, let's say, area under the curve 2 to area under the curve 4. And similarly, it would also be linear in the brain. So the binding potential the first time would be 16 over 2, which is 8. The binding potential would be 32 over 4 the second time. It would still be 8. Just because we've doubled the dose, we haven't changed the density of the serotonin transporters in this subject, of course. So it corrects for injected dose, and it also corrects for body weight, because if it's distributed to other portions of the body, it will be lower concentrations in the plasma. And it also corrects for faster metabolism, because if there's faster metabolism, this will come down faster, and the area under the curve will be lower. So that's how you do it. Okay, so the major point of PET pharmacokinetics in words can be done this way. The plasma pharmacokinetics provides a limited view of what's happening to the drug in plasma. And you will receive many lectures on pharmacokinetics. And what they are talking about is the concentration of drug in the blood over time, kinetically. But that's a very limited view of what's happening to the drug. PET provides a limited view of what's happening to the drug in the tissue, in my case, in the brain. But if you concurrently measure the drug in the plasma and drug in the tissue with PET, you can quantify the target of drug action, that is the receptor. Okay, so pharmacokinetics, as I mentioned, is the study of the drug in plasma. And that is often distinguished from pharmacodynamics, which is like the effects that the drug has after it binds to the receptor. So the dynamic effect of a drug to lower blood pressure, what effect did it have on blood pressure? But the very first pharmacodynamic effect is when the drug binds to whatever receptor, the alpha-2 receptor or whatever, that this antihypertensive agent uses. So we are measuring the very first step of pharmacodynamics, and that it's binding of the drug at the receptor. And the combination of those two allows us to measure receptor density, which can be important if it's related to disease. So the plasma and the drug is shown in that slide. The drug in the plasma is shown in the first slide. The drug in the brain is shown in the second one, and we can measure receptor density. And so this can be used quantitatively to measure the site of drug action, whether it's the dopamine transporter in Parkinson's disease or the amount of amyloid in a patient with Alzheimer's disease. Okay, so that was an intuitive understanding of what pharmacokinetic modeling is. Let me show you now how PET can be used to study distribution. And again, distribution in the pharmacological sense refers to the reversible transfer of a drug from the plasma to the tissue and back. So if it goes to the tissue and never comes back to the plasma, it's been eliminated. But if it goes and comes back, goes to the tissue and comes back, that's distribution. And I'll do this with regard to a receptor previously called the peripheral benzodiazepine receptor. Okay. The current name for this is the translocator protein 18 kilodalton. It is also known as, or previously known as, the peripheral benzodiazepine receptor. It is a mitochondrial protein that's highly expressed in macrophages and activated microglia, which are phagocytic inflammatory cells. The microglia are in the brain, macrophages are in the periphery, but they're very, very similar cells. It exists in both the periphery and the brain. It was first discovered in the kidneys, but then they found it was in the brain, so that's why they had to take peripheral benzodiazepine receptor out of there. It has multiple potential functions. One that's most talked about is steroid synthesis, but I'm not going to get into its function. I'm just going to use it, but I do want to say it is distinct from the typical benzodiazepine receptor, the Valium receptor, which is the GABA-A receptor in the brain. This is not the GABA-A receptor. This is a transporter protein on mitochondria, highly expressed in phagocytic cells. We're using it really just as a marker for cellular inflammation where these cells are present in localized or generalized inflammation. Okay. So we developed a radioligand, a PBR28, that binds to this TSPO. And we evaluated it. And these are the typical sort of curves that I was mentioning to you before in the brain and in the plasma. So this is the concentration of this radioligand in the monkey brain over time. And then this is the concentration of the radioligand, parent radioligand, in arterial plasma. And note that this is a log scale. So it's declining very rapidly here, but it's still staying in the brain because these receptors have high affinity for the radioligand. In this case over here, before the animal was injected with tracer doses of PBR28, the animal was injected with pharmacological doses of a non-radioactive drug that also acts at this receptor. And you can see in this case that there's a much different curve. So there's an early and higher peak. It's rapidly washing out of the brain. And in the plasma, the concentration is much, much higher. Like this is almost 10-fold higher. Okay. Well, how can we understand this kinetics? The baseline condition, what's happening is that the drug, when you have a high affinity binding site in the brain or in any organ, the drug binds and comes off, binds and comes off, binds and comes off, and it sticks there. It's like a magnet that is able to keep these iron particles, the drugs, closely associated with it. And it stays there in the brain much longer than in the plasma. When we gave the receptor blocking dose of a non-radioactive compound, if the non-radioactive compound has gotten on there and blocked the binding of the radioligand, it's sort of like that children's game, is it Goose Goose, Duck Duck Goose, or whatever it is, in any case, where you have to get up and one chair is removed, and if other people are occupying it, then you're not able to get on there. And that's what's happening here. The reason why it's rapidly coming out of the brain is that you've blocked all of those sites. You've sort of demagnetized the magnet, so it no longer has an affinity. It's not going to stay in the brain. It's going to wash out of the brain as quickly as it washes out of the plasma. So we can understand the basic pharmacokinetics of this, why it's washed out much faster. But why is there so much more in the brain? Why did this go up to 500? Well, the reason for that is because there was more in the plasma. Actually, there was 10 times more in the plasma, so that's why there was more transiently in the brain. Well, why was there 10 times more in the plasma? Because actually we injected the very same dose in the identical monkey. Aha. That's the issue of blocking peripheral distribution. And I can show it to you here. Oh, but before I move on, if you did that calculation that I mentioned before, you did this, extrapolated to infinity, you did this and extrapolate to infinity, and took the area in the brain divided by the area in the plasma, the number that you get would be 130. If you do it on the blocked scan, the number you would get would be 1.7, which means that more than, this is an outstanding radioligand in the monkey brain, more than 99% of it is specifically bound to receptors because we can displace it with other pharmacological agents from that site. So the vast majority of the uptake in the brain is reflecting binding to the TSPL. Okay. Receptor blockade displaces a radioligand in the lung and the kidney and drives more to the brain, but it doesn't stick in the brain. So I've been showing you images of the brain, which you can see in this whole body monkey scan. That's all that we're measuring. But in the periphery, there are huge amounts of this TSPO. It was first discovered in the kidney and also in the lungs and in the heart and the spleen. And when you give the blocking dose, you not only block in the brain, but you block in the periphery where there's huge numbers of these sites. So the non-radioactive drug, PK in this case, blocks distribution in the periphery, leading to much higher concentrations in the plasma, 10 times higher concentrations of the radioligand in the plasma, which transiently go into the brain, this initial blush, but then wash out because the magnets have been demagnetized. So this is an example of showing how PET can measure distribution, in this case by blocking the distribution to peripheral organs that have a very high density. You can transiently deliver more to the brain. Okay, I'd like to take a little digression here in two ways. First is, here's an example of a regular, healthy human subject here. And this is the brain time activity curve. And this is a normal monkey. So which do you think, by the way, has more TSPO? A normal monkey or a normal human? Well, because there was much more uptake that stayed a longer time. It's basically, we've done the full quantitation, and the monkey has 10 or 20 times as many TSPO as a human healthy. And this was the example that I showed you before of a pre-blocked monkey. There was higher uptake, and then it washes out very quickly. Well, we came across this odd, healthy human subject who had a higher uptake. Notice the difference in the scale, and it washed out immediately. And it looked like a pre-blocked monkey. So given the fact that this was a young male in his 20s in the United States, we said he's probably on drugs, and the drugs must be blocking it. But he said no, and his drug urine tox test was negative. But he did say he was taking quite a bit of ibuprofen, Motrin. And we thought, well, maybe Motrin's binding to the site. He stopped taking it, we came back, and sure enough, he had the same thing, rapid washout of activity from the brain, as if he had no TSPO. Well, we said, well, if he has this receptor in the brain, maybe he has none of these receptors in the periphery. And here's a whole body image of this TSPO radioligand. Like in the monkey, there's very high uptake in the lungs, the kidneys, and the spleen. But in this no binding subject, which represents about 10% of the healthy population, there's no binding there. It turns out, to make a long story short, that these individuals do have TSPO, but they have a single nucleotide polymorphism. A single nucleotide polymorphism, which causes this differential affinity. And that causes a substitution of an alanine to a threonine in the TSPO. The allelic frequency of this is about 30%, and therefore the presence of the homozygote, you have two genes, 30% of 30% is about 9%, so about 10% of the population. And you have individuals who have too high affinity binding, too low affinity binding, or actually a combination of the high and the low. But the low affinity binders represent about 5 to 10% of the population we're finding in this area. So this is a case where there are individuals who have different affinity for the radioligand. This is quite unusual, and it's caused by a single nucleotide polymorphism. We can still use this radioligand, but we have to correct for, either statistically or by matching, the genotype of the individuals. Well, if you do that, I want to show you how this marker of how PET can be used to study pathophysiology, in this case of TSPO. So this marker of inflammation, highly expressed in activated microglia, which are sort of the macrophages of the brain. Neuroinflammation is known to exist in Alzheimer's disease at the time of death, massive neuroinflammation. And it may be a contributor to the disease pathology, But it's unclear whether the Alzheimer's disease, whether the neuroinflammation begins early or late in the disorder. It's present at the time of death. Prior TSPO PET studies have shown conflicting results in Alzheimer's disease and also in mild cognitive impairment, which is the precursor syndrome, a little bit of memory problems. This PBR28, I won't go into it, but is a markedly improved, much greater signal-to-noise ratio, much more specific binding than the traditional one. So we used it with the genotype correction, as mentioned, to look for expected differences in TSPO density between Alzheimer's disease and controls, and also MCI, which we didn't know how they would turn out to be. The bottom line is shown here, published in 2013, I think. Basically, there's increased TSPO density, and this is a mean image of a number of subjects. It's increased in Alzheimer's disease, but not in mild cognitive impairment compared to controls. I think one of the most important things that might have been expected, but this was not necessarily expected. So this looked at the correlation between the binding and the disease severity, a clinical dementia rating scale. And when on a standardized X and Y axis, there was a very strong correlation. So it means cross-sectionally between individuals, the more inflammation you have, the more cognitive impairment you have. So it appeared to be a biomarker of disease severity. And that can be understood in some ways in this progression of various aspects in Alzheimer's disease. So there are various markers or functioning that are shown here, and this is generally accepted. The very first thing to develop in Alzheimer's disease is actually the A-beta, these plaques of amyloid beta. And they reach a peak just about at the beginning of MCI and stay about the same. So it had been known from postmortem and also from imaging, PET imaging studies of amyloid, that there's no correlation between disease severity and the amount of amyloid. And the reason for that is that the amyloid reaches a peak very early and stays at that high level. While the patient, these are other markers, here if you look at clinical function, this one over here, it's deteriorated just a little bit in MCI, and then it goes to hell in the dementia. Well, the dysfunction is increasing, but the amyloid is staying the same. So that's why there's no correlation between amyloid and function. What we see with the neuroinflammation is more similar to this here. It's a marker of the transition from MCI to dementia, and that it correlates, the more inflammation you have, the greater the cognitive impairment. We also followed this up with a longitudinal study in some of these individuals to determine if the TSPO bind increased during the progression of Alzheimer's disease, and also compared it to normal aging. And subjects were scanned 2 to 4 years, were scanned again 2 to 4 years after the initial scan. And what we found is that the PBR28 binding increased in the patients, in every patient, and there was no change in controls over this 2 to 4 year period of time. The individual subjects are shown here. Every subject over this 2 to 4 year period of time increased in this neuroinflammation, whereas the controls, there was no increase whatsoever. But again, arguably the most important thing is the correlation here. The increased PBR28, the amount of increase in the binding, correlated with the amount of increase in clinical severity, with the change of this cognitive marker, the sum of boxes squared. And the correlation was really quite strong. So this would suggest that neuroinflammation is a biomarker of disease progression. To summarize that here, a cross-sectional study in which everybody got one PET scan. Neuroinflammation occurs after the conversion of MCI to AD and worsens with disease progression. It is therefore a putative biomarker of disease severity. The longitudinal study showed this TSPO labeled with PBR28 increases in Alzheimer's disease but not in controls, and that the amount of increase correlates with disease progression, and therefore it is a putative biomarker of disease progression in Alzheimer's disease. These are small pilot studies that need to be replicated in other centers, and I think largely they are being replicated. So this shows how a marker of inflammation can be used in ways that sound pretty important, disease severity and disease progression, when you consider therapies that might be tried. And anti-inflammatory therapies are worth, I think, reconsideration in patients with Alzheimer's disease. Okay, to summarize then, I use TSPO as an example of studying distribution, in this case between the periphery and the brain, and how if you block both of them, you can leave more you can have a higher concentration in the plasma, and then lead transiently higher levels to distribution to the brain. I also wanted to show that this marker itself could be useful in Alzheimer's disease, and in many others we're looking at inflammation in either the brain or in the periphery. And now I want to take another little digression in talking about distribution for one study that we did that I thought was really very fascinating, and it's PET imaging of PGP, permeability glycoprotein. So this is looking how PET can show distribution, in this case blocking the distribution. It is an efflux transporter, which I'll explain, which for the brain at least helps to protect the brain from toxins, but it can also cause drug resistance. Permeability glycoprotein, or PGP, for which there may be a separate lecture later by Michael Gottesman, it transports drugs out of cells in many locations, for example, in the brain and in the testes, so that drugs, let's say potentially toxic compounds, are not able to accumulate in sensitive organs like the brain and the testes. With regard to the brain, it is a specific component of the blood-brain barrier. The general idea of the blood-brain barrier is that there's the endothelial, the capillary walls are very tightly linked to each other. And so in order for a drug to get in, it has to go across a lipid bilayer. So it has to be lipophilic. That's considered like the structural component. But there are other blockers at the barrier, including these efflux transporters like PGP that I'll show you. And an example of a substrate for PGP is loperamide, Imodium. Imodium is a potent opiate that acts on the gut to slow motility, but it has no actions in the brain whatsoever. So there are opiate receptors in the gut, and when the opiate binds to it, the gut will move more slowly, and therefore there's more time for the water to be taken up. And if you have diarrhea, you can take loperamide, but you won't get high. If you're a heroin addict, one of your common complaints will be constipation because you're taking too much of it. Okay, the reason why you don't get high is because PGP blocks the entry of this opiate, but not others, in the brain. And I should mention that it is overexpressed in half of tumors that are resistant to chemotherapy. So these drug-resistant tumors, maybe half of them overexpress PGP and other efflux transporters, which in some cases block the entry of the cancer chemotherapy. And it also occurs very likely in the brain for drug-resistance epilepsy, where there's overexpression of PGP, blocking the entry over time of years of treatment of the anti-epileptic drug. So it helps protect the brain, but it can also block the entry of therapeutic compounds like anti-epileptic drugs, likely causing drug resistance in epilepsy, which is about a third of the patients. Okay, so this diagram, which is a takeoff from one that Michael Gottesman created, looks at the blood in a ring on the outside, and then you have the tissues here, and then you have the excreted areas here. And it looks at the distribution of PGP, which is throughout the body. As mentioned, PGP is located at the blood-brain barrier in the capillary endothelium, and it will transport drugs out of the brain. It's also at the testis barrier and won't let drugs like loperamide or Imodium go into the testes. It's overexpressed in multi-drug resistance tumors, MDR tumors. But it's in other examples also. So in the placenta, for example, it'll block the entry of drugs that are substrates so it would protect the fetus. Okay. So it's widely distributed. P-glycoprotein removes lipophilic substrates directly from the plasma membrane. So if this is the blood-brain barrier, blood-testis barrier, this out here would be the blood, and this inside here is the brain. And the substrate, loperamide or whatever, any substrate, it begins to cross the membrane, but then PGP captures it and excludes it. So it actually, if it's a very potent substrate, the drug itself will never even make it into the brain. Instead, PGP will pump it immediately out before it even gets through the lipid bilayer. So we wanted to measure the function of this efflux transporter, and we made a radioactively labeled loperamide. It was the D doesn't have a methyl group on it. For various reasons, it's better. And you can see that in a normal brain, in a wild-type brain of a mouse, there's no uptake in the mouse brain. But if you look at a PGP knockout mice that doesn't have it, then there'll be uptake in the brain. So these mice will get high, and do get high, from loperamide, but if they're knocked out, but not if they're wild type. And if you look at the amount, it really makes quite a big difference in the brain. The wild type has hardly anything, but the PGP knockout has a lot getting into the brain, so it's getting high. Going up the phylogenetic tree, this looks at the uptake in a monkey brain. This is a coronal section through a monkey brain. And here's the pituitary for reference. We don't have a knockout monkey, but we can give a drug that will block PGP. And we see that if we block the PGP, the drug is allowed to get into the brain, and the monkey would presumably be getting high. We've also used and the amount of uptake is really quite striking. So there's very little uptake of the radio-labeled loperamide at baseline, but if you block the efflux transporter, a huge amount, manyfold, can get into the brain. So this is showing in animals that this is an appropriate substrate measuring the function of PGP to block the entry of the drug into the brain. Here's the distribution of C11D-LOP in a healthy male. And it looks at various times, looking at the radioactivity, at 3 minutes, 20 minutes, and 100 minutes. On this here, you can see, well, there's high uptake initially, like in the kidneys, the lungs, and the thyroid. And the reason for that is that they have a high blood volume. There's a high amount of blood in each of those organs. So you see initially sort of the blood distribution with the drug in the blood. At later time points, you can see metabolites or potentially binding to PGP sites there. I know when I showed this some years ago to some females in the lab, they said, oh yeah, this is a typical male. No brains, no uptake in the brains, and no uptake in the testes. Typical male, no brains, no balls. And that's true, because the PGP blocks the entry into the brain and into the testes. Okay. Looking at this early summed images from 0 to 3 minutes, you see a lot of the blood pool. So let's see if this image would go around. First, before I start this rotating, this is the site where it was injected, and there's high uptake because it's a lipophilic compound, in the endothelium of the vein as it comes in. Here's the thyroid, kidneys. Let's take a look at that. But look in the brain. Well, there's this. This is for whatever reasons, which I don't really fully know. There is high uptake in the pituitary, but you also see this structure here. And I wonder if any of you know what that is. That is the venous sinus of the brain, and which we hardly ever see in the PET imaging studies that I do. This drug, as I mentioned, the radioligand, is blocked from entering the brain, and it stays in the blood. And the blood of the brain is drained. The venous drainage goes into these very large veins, so big that they are called sinuses. So what we are seeing here is that we're able to see the blood in the sinuses as the draining veins of the brain, and we can see it because there's no uptake in the brain. Most of the radioligands that I develop go into the brain, so therefore we can't by contrast see it. Okay. Alrighty, so this looks at a fused image. This is the MRI of, let's say, that individual. You can see that there's very high uptake in the pituitary for reasons that I won't go into. But there are some other areas of high uptake. But the uptake, this number is very, very low if you look in the brain itself. In addition to the pituitary, there are these other structures that are labeled. And if anybody out there can tell me that one, I'll give you a case of Heineken. Okay, so here they are in another distribution or different tomograph. What are these sites here, which are located in the medial surface of the lateral ventricle, which are located in the roof of the third ventricle, and are also located in the roof of the fourth ventricle? The ventricles are the CSF drainage from the brain. And the answer is that this is the site where there's the drainage of CSF from the brain. Can you take one break? Why am I blocking it? Okay, so this uptake, I'll go back one. So these uptake is in a tissue in the brain called the choroid plexus. The choroid plexus is a tissue that is involved with producing the CSF, the cerebrospinal fluid, or the water in the brain. And for whatever reasons, drugs that are substrates for PGP also happen to accumulate in the choroid plexus, which I have never seen before. I've seen the ventricle maybe a little bit. So here's an image of it. You can see the venous drainage, the venous sinus, but here now, three-dimensionally, you can see better this choroid plexus, which is the tissue that makes the CSF for the brain, the water in the brain. Okay, well, can we increase the uptake of a drug with a PGP inhibitor in human subjects? And we can. So you can see this is the baseline uptake, no uptake in the brain, but there's uptake in the choroid plexus. And if we give this inhibitor at 6 milligrams per kilogram, we can increase the delivery of the drug to the brain. And that's shown here. The brain uptake is increased with this inhibitor in a human just like it is in a monkey. So it suggests that for those tumors that could have increased PGP, you could potentially increase the uptake of the cancer chemotherapy agent into the tumor, or you could increase the delivery of the drug to the brain with a PGP blocking agent. In any case, I found this a very fascinating study to show how PET could study an important regulator of distribution, in this case the PGP efflux transporter, which blocks the entry of those drugs which are substrates in many tissues in the body. Okay, so I gave some examples of how PET can study the distribution. In the case of the TSPO, previously known as the peripheral benzodiazepine receptor, you can look at the competitive distribution or distribution to the brain and the periphery. You can also look at how the distribution of drugs are blocked by efflux transporters, including PGP. I want to end with showing how PET can also study drug metabolism. And metabolism, again, is where you make or break a bond in the drug. You change the chemical structure. This radioligand, FC-Way, is one that was developed at NIH, and it labels a particular type of serotonin receptor in the brain. But at 2 hours after injection, the vast majority of uptake is just in the bone, the skull, surrounding the brain, but very little uptake in the brain. And the reason for that is that FC-Way is relatively rapidly metabolized. It's defluorinated so that you end up with F18 fluoride ion, and, as I showed you previously in that rat study, taken up into the bone, including into the skull. So this is the structure of Fc whey, which is defluorinated, and you end up with fluoride ion here, which is taken up in bones of the body, including the brain. But it obscures the picture, and there'll be some spill-in of activity from the skull into the surrounding neocortex. So one of the mechanisms of defluorination is a cytochrome P450 enzyme, CYP2E1, if I've gotten that correct, is a defluorinating enzyme in the liver. So we wanted to see if miconazole, which is known to inhibit this enzyme, would be able to inhibit radio defluorination and decrease bone uptake. Miconazole is an antifungal agent. It's also advertised as quick-acting tenectin, and it's a potent inhibitor of CYP2E1. This looks at the uptake of fluoride ion if a rat is injected with F18 fluoride ion. In this case, down below, we've injected F18 Fc whey, and initially there is a lot of uptake in the skull, particularly the base of the skull, which is thick in a rat and in a human. But then in these studies, we gave increasing concentrations of miconazole, and it could decrease the uptake into the skull and give you more delivered into the brain. And this is the hippocampus of a rat, which has the highest density of this particular type of serotonin receptor. We wanted to see if we could do this in human subjects, but we couldn't give miconazole intravenously. Instead, we used disulfiram, which is known as antabuse. Disulfiram, I think, is the most potent inhibitor of CYP2E1. It inhibits many different enzymes, and it is used clinically to maintain sobriety, far more frequently in Europe than in the United States. But if you take this drug, which inhibits the intermediate metabolism of alcohol, you have a buildup of an intermediate metabolite that makes you flush and gives you tachycardia, it makes you sick and nauseous. So if you take this disulfiram every day, you're not going to take a drink because you'll get sick. So we gave the disulfiram the standard sobriety dose, 500 milligrams on the night before, and it was just a dramatic effect. We could block the defluorination, block the uptake in the skull, and deliver more into the brain. So here are the time activities for that. You can see that disulfiram, looking at it in the skull, just virtually blocked all the uptake in the skull because it was blocking the defluorination. If you look in the brain, it really delivers far more to the brain. It's not metabolized, so therefore there's more radioligand in the plasma to go into the brain and bind to these serotonin receptors. If you look at the concentration of fluoride ion in the plasma, the disulfiram almost completely blocks the amount of fluoride ion that gets there. And then if you look at the parent tracer, because you're blocking the metabolism, there's more of the parent compound, and there's more that would go into the brain. So this shows how you can use PET to study the metabolism, in this case of a radioligand, and in this case of the defluorination. It turns out that defluorination is fairly common. There are different mechanisms in which it occurs and can be a problem for F18 labeled drugs. If it occurs via CYP2E1, you can eliminate that problem by giving the subject just the standard single sobriety dose of disulfiram. Let me summarize then what I've told you today. PET, positron emission tomography, is a technique that has very high sensitivity and specificity. It has high sensitivity, meaning that it can measure that radioactive atom, carbon-11 or F18, at very low concentrations, 10 to the minus 12th, 10 to the minus 14th molar. Therefore, it has adequate sensitivity to measure many of the proteins that we're interested in, which are present at 10 to the minus 9th molar. It has specificity insofar as if the radioactive atom is connected with a drug that binds specifically to the D2 type of dopamine receptor, then you'll be labeling specifically that receptor. PET is used in therapeutic drug development currently in several ways, perhaps most commonly in helping to determine the initial dose of the drug, because on the initial dose you don't want to give too much and have unnecessary toxicity, and you won't want to have too little and miss the potential efficacy if you had used the proper dose. Pharmacokinetic modeling of PET radioligands involves simultaneously knowing the concentration of the drug over time in the plasma as well as in the tissue. And having both of those together, a pharmacokinetic measure and the very first of the pharmacodynamic measures, you can, in combination with that, find out the density of the target site of the receptor in the tissue of interest. I've shown how PET can study distribution, which is the reversible transfer of a drug from the blood to the tissue and back. The case I gave of translocator protein was one where the receptor is located in the brain and the periphery, and you can have sort of a competitive distribution of the drug or the radioligand to those two sites. I also showed that that particular target can provide useful pathophysiological information on inflammation or neuroinflammation in my particular case. And finally, I showed how PET can study metabolism, making or breaking a bond in the radioligand. And in this case, it was the defluorination and the ability to inhibit the defluorination and have more of the parent radioligand. Let me finish up, and let me remind everyone who's taking this course for credit, this is the time to wake up. I'm going to give you all a self-assessment quiz. Try to remember this. True or false? Let's go over them. Imaging with positron emission tomography, PET, involves the injection of a radioactively labeled drug that emits a particle called a positron. And the answer is true. That's true. The particle is released, the positron. It later will annihilate with an electron, matter and antimatter combining, giving off two gamma rays that are high enough energy to go straight through the skull and can be measured with a PET camera. PET shows the location of radioactivity in a cross-section or tomograph of the body. Yes, that's what the T in tomography comes from. A tomograph is not just a flat image, but it's a slice going through. We have the front and the back and the left and the right. So that is true. PET can be used to quantify the density of specific proteins in the body. The answer to this is also true. If you simultaneously measure the concentration of the drug in the plasma and in the brain, let's say, then you can determine how many receptors are there. That is, you'd have more uptake in the brain relative to how much was delivered in the plasma. Okay, compartmental modeling, which is very complex, of PET data typically uses the measurements over time of PET images of the target tissue and concentrations of the unchanged parent radioligand in plasma, and the answer to that is true. It has pharmacokinetic measurements in the plasma and the initial pharmacodynamic measures in the tissue, and with those being measured simultaneously and extrapolated to infinity, you can get a measure of the density of the target in the body. Thank you very much for your attention. I hope I was adequately able to communicate my enthusiasm for this technology, positron emission tomography, which I think can be useful for studying the brain and other organs in the body. And it's based upon pharmacology. So be well, do good work, and keep in touch.